My name is Dr. Ronan MacLoughlin, Associate Director of R&D Science and Emerging Technologies, and I'm here to present the on-demand presentation entitled The Assessment of Aerosol Drug Delivery During the Escalation of Treatment for a Simulated COVID-19 Adult Patient. In terms of disclosures, employed by Urgent, design and manufacture of five-ring nebulizers relating to five-ring mesh nebulizers and their applications. And we are currently engaged in 15 plus prophylactic and therapeutic interventions for COVID-19. None of these I will go into any detail whatsoever in this presentation, but the methods and the testing that I describe later are similar to those approaches that we've taken in terms of de-risking clinical trial design, optimizing dosing strategies and dosing formulations. I think at this point, nobody needs to hear any more about COVID-19 or the SARS-CoV-2 virus, the respiratory virus that is the cause of COVID-19 that has led to more than 5 million deaths globally over the last year and a half or thereabouts. It has interestingly led to a resurgence in interest in respiratory drug delivery as a means of clinical and therapeutic intervention. And more recently has been seen as a route of administration for prophylaxis, such as monoclonal antibodies, but also vaccines, with some interesting and very promising data published very recently with collaborators that would indicate that inhaled vaccines have a potential place in the future of respiratory care and prophylaxis for COVID-19 and other infectious diseases indeed. With respect to COVID-19 patient and the escalation of care that they may require, generally speaking, the COVID patient is diagnosed as COVID patient in the home care setting. Hopefully, it's where they'll stay and symptoms will be mild or they'll be asymptomatic. But as the patient progresses and gets sicker in those instances, they will end up requiring clinical intervention in the clinical setting. Each of those patients will be sick and require a variety of different patient interventions depending on their own clinical requirements. It's generally limited to oxygen supplementation and various different respiratory supports. But in this figure taken from Fernandez et al., a paper published earlier in 2021, you can see that the various different patient interventions vary from emergency room ED to the ward to the ICU, where you'll have spontaneous breathing with low flow or high flow nasal therapy. We've gone to more invasive or more complex interventions such as non-invasive ventilation in the wards, or in the ICUs you have the more invasive mechanical ventilation where the airways are instrumented with an endotracheal tube, non-invasive ventilation, or indeed high flow nasal therapy, which has become exceptionally popular during the COVID-19 pandemic. We will all be familiar with the spontaneous breathing patient inhaling their dose via the mouthpiece or face mask. However, high flow nasal therapy and high flow nasal cannula are increasingly being used as a means of aerosol drug delivery concurrent with oxygen supplementation of the hypoxic patient, with several clinical trials looking at concurrent aerosol drug delivery and the effective bronchodilation achieved by the drugs delivered during high flow therapy. Obviously, as the patient becomes more and more hypoxic, you will end up, hopefully not, but you will end up requiring more invasive ventilation interventions that require the patient to be sedated, instrumented, and the machine will do the breathing for that patient. Now, the research question we tried to address during the study was, indeed, what is the effect of those different patient interventions on the amount of aerosol delivered to the patient, the COVID-19 patient more specifically, and how does that compare to what we know in terms of current state-of-the-art published in the literature, which is generally published along with international standard breathing patterns that will be stated in the likes of the FDA guidance document for nebulizers, PMDIs, and DPIs, or indeed ISO 27427 and similar standards. The implication of this is becoming more and more obvious over time. This recent study published by Nocton et al. Would suggest that in the low tidal volume ventilation strategy that is adopted for the COVID-19 patient, that the choice of device, aerosol generator device, can have a significant effect in terms of the mass of drug delivered to the patient's lungs. What isn't yet known is the question what we're trying to answer here in terms of the effect of the patient intervention that's prescribed to the COVID-19 patient. Now, the implication and the importance here is obvious in that if you search clinicaltrials.gov, nearly 10% of all trials registered for COVID-19 are actually in inhalation trials. So there's just over 600 trials looking at inhalation COVID-19. Many of those trials will be using aerosol generators such as nebulizers, pressure controls, inhalers, etc., etc. And the influence of the patient intervention across that broad range of patient types, COVID patient types, will have a profound effect on the clinical outcomes. So in an effort to address that gap in our knowledge, we sought to apply standardized materials and methods using spontaneously breathing patient models, such as those described in previous papers of ours and others, looking at using breathing simulators for patients for spontaneous breathing via mouthpiece, via nasal cannula, and also invasive mechanical ventilation setups. As I mentioned, we're comparing standard normal adult breathing patterns that we'd all be very familiar with, and comparing that with mild COVID-19 adult breathing pattern. This breathing pattern of 25 breaths per minute, tidal volume of 600 mils, and an IE ratio of 1:1 is a composite of several different mild COVID patients that we have taken from recordings from various different institutions around the world over the last 18 months. The nebulizer used was the Airgen Solo vibration mesh nebulizer. This was not a device head-to-head comparison. The reason the Airgen Solo was chosen was it's the only device suitable for use across each of those patient interventions. The high-flow nasal therapy system used was the Fisher & Paykel Airvo at 30 and 60 liters per minute. The invasive mechanical ventilator was the McKay Servo I, used in combination with a HME, a heat moisture exchanger, and it followed lung protective ventilation strategies described in the ARSNet protocol at 20 breaths per minute, tidal volume of 400 mils, and IE ratio of 1:2. This is considered a lung protective strategy where it delivers lower volumes to the airways in order to avoid VILI, which is ventilator-induced lung injuries, and is an accepted and widely adopted method for ARDS patients and consequently COVID-19 patients. The results of the study are illustrated in front of us, where we can see for the spontaneous breathing patients, the healthy and the mild inhale dose using the Airgen Ultra device was pretty similar in that there was no significant difference between the two. This may indeed have been a function of the fact that the Airgen Ultra device acts as a spacer, where a bolus is inhaled on inhalation, and during exhalation, the exhaled breath bypasses the chamber wherein the bolus is building up, ready for the next inhalation. During high-flow nasal therapy, the 30 and the 60 liters per minute gas flow rates both recorded similar data, somewhat lower than we would have expected for other breathing patterns, such as the normal breathing pattern, and again, lower than what we've seen for a distress breathing pattern published by Romaniak some years ago. For mechanical ventilation, however, we've seen the more profound and more significant difference between the delivered dose between the published literature, which is the normal breath or the standard breathing patterns, versus the LTV, the low tidal volume ventilation parameters. In this instance, the reported 30% or thereabouts for standard breathing pattern on mechanical ventilation was nearly halved to approximately 15% lung dose. Now, this is important on the basis that any therapeutic, any research that would have been done looking for an understanding for how much a ventilated patient may receive would have significantly overestimated that dose on the basis of the literature. This, for the first time, gives an indication of how little, perhaps, or how significantly less that dose is compared to the published literature, and may indeed have a profound effect in terms of clinical outcomes for the COVID-19 patient. As I mentioned, the results can be summarized to say that patient intervention has a significant influence in terms of the delivered dose to the patient's airways. It does not seem to be using the Airgen Ultra, at least in the spontaneous breathing patient via mouthpiece, any significant difference between normal and mild COVID breathing pattern, nor does there seem to be any difference between 30 and 60 liters per minute in the COVID patient. And as discussed, the lung protective ventilation strategy did indeed have a significant effect. The implications of this data would suggest that there is indeed the potential for massive dose variability across patient intervention. And this is a key point to take home on the basis that COVID-19 patients will either progressively get worse or hopefully progressively get better, but they are not the same patients. And should a drug be delivered to that patient under different patient interventions, they will be getting an altered dose, and that potentially compromises the readouts from clinical trials when there's variability in drug delivery simply because of the patient intervention. And now, for the first time, we do have more of an understanding of what that potential dose might actually be in the mild COVID patient, whether that be the spontaneously breathing or indeed the mechanically ventilated patient. So with that and the 10 minutes that we have, I sincerely thank you for your attention. I look forward to any questions that you may have. Please feel free to email me at the address provided, and I look forward to hearing from you. Stay well, stay safe.